everybody. Hope you all are enjoying good health. Today we are going to start a series of 3 lectures about gametogenesis. The first session comprises of general features of gametogenesis, and the participant at the end of the lecture should be able to give a brief review of theory of inheritance, give the basics of how to assess human genome and its abnormalities, describe the process of gametogenesis, give a brief review of hormonal regulation of gametogenesis, and finally give a review of the clinical significance of gametogenesis. In second session, we will discuss the process of oogenesis and its clinical significance, whereas in session 3, we will discuss the spermatogenesis with its clinical significance. Let us start with the theory of inheritance. Chromosome is a microscopic thread-like organized structure of DNA compacted by proteins. It carries hereditary information in the form of genes. And the chromatid is a newly produced copy of a chromosome still attached to the original chromosome by a single centromere. Gene, a basic functional unit of heredity, which is transferred from parent to an offspring and is held responsible to determine some characteristics of the offspring. Humans have about 23,000 genes on 46 chromosomes. According to Stuart, genes arrange in serial order on chromosome and tend to be inherited together or named as linked gene. And these similar genes present on similar loci on a homologous pair of chromosomes are termed as allelic genes. Chromosomes having genes are used as vehicles for genetic heredity, and their behavior involves segregation, independent assortment, and occasionally linkage. And recombination between homologous can occur anywhere on a chromosome with equal likelihood. In a newborn, traits are determined by specific genes on chromosome inherited from both the parents. Karyogram is a graphical depiction of complete set of chromosomes in a cell. A somatic cell have got 23 homologous pairs of chromosomes, and the cell is termed diploid, having 46 total chromosomes. Out of these, 22 pairs are matching chromosome, which are named as autosome, and one pair is unmatching, termed as sex chromosomes. If sex chromosome pair is XX or double X, individual is genetically female. Whereas if the sex chromosome pair is XY, individual is termed as genetically male. One chromosome of each pair is derived from maternal gamete, the oocyte, and the other chromosome of that pair comes from paternal gamete, the sperm. Thus, each gamete has a haploid number of 23 chromosomes. which on union of gametes at fertilization restore diploid number of 46. Human genome and its assessment. Genome assembles complete set of gene in a species, and genotype is total complement of both the autosome and sex chromosomes. 46XY is termed male, whereas 46XX is female. Assessment of the human genome is accomplished by karyotyping, in which cells are lysed and chromosomes assessed microscopically with the aid of fluorescence tagging. This process gives a likelihood of detecting any chromosomal abnormality. Now, how this process is accomplished? Fluorescence in situ hybridization is performed by using a probe for chromosome 21 as indicated by red dots in the lower diagram. Notable things in this diagram are: number one, there are three red dots in each cell which indicate trisomy 21, that is Down syndrome, and the green dot, one green dot in each cell, is the control probe for chromosome 13. There is an artifact in the right lower corner of the diagram, which is because of superimposition of two cells, which give impression of presence of multiple probes in that cell. Now, gametogenesis. It is a delicate, specialized biological maturation process in which formation and development of specialized reproductive cells, that is gametes, are obtained from the primordial germ cells. This process is mandatory for the propagation of species and is accomplished in two steps. Number one, reduction of chromosome number and rearrangement of genetic material. Number two, acquisition of special form and function by the cells which prepares them and make them suitable for fertilization. These characteristics are acquired in two stages. Number one is meiosis, which comprises two parts. In meiosis 1, there is reduction of chromosome number through the process of disjunction and rearrangement of genetic material through crossover process. In meiosis 2, there is reduction of genetic material through independent assortment of chromosome among the newly produced cells. Number two is cytodifferentiation, in which complete maturation of the newly formed cells takes place, and this is more seen in the spermatogenesis process. Gametogenesis hormonal regulation. In both male and female, reproductive cycles are controlled by interaction of hormones from hypothalamus and anterior pituitary gland with the hormones produced by the respective reproductive organs in each sex. In both sexes, hypothalamus monitors and through releasing gonadotropin releasing hormone causes release of hormones from pituitary, follicle stimulating hormone and luteinizing hormone. These two hormones acting on gonads, that is ovaries and testes, produce estrogen and testosterone respectively. All these hormones play important role in controlling reproduction through gametogenesis in the production of ova and sperms. As regards the clinical significance, gametogenesis is responsible for constancy of chromosome number from generation to generation by producing haploid sex cells, and the diploid number is restored at the time of fertilization. Number two, genetic variability is enhanced during gametogenesis through number one, crossover process which permits mixing and recombination of genetic material, and number two, independent assortment that allows random distribution of homologous chromosome to the daughter cell. This variability is essential for evolution. As earlier said, there are two types of gametogenesis. Number one is oogenesis for the production of ova, and second one is spermatogenesis for the production of sperms. These two topics will be discussed in the coming sessions. Comments and suggestions will be welcomed. Thank you for listening and watching.